Joining me is Dr. Eva Chi, Assistant Professor of Chemical Engineering at the University of New Mexico, doing research in the area of protein dynamics. And more specifically, I guess one of those topics, Dr. Chi, is Alzheimer's. What is preventing us from finding a cure?
We don't yet have a good understanding of what causes the changes that we see in Alzheimer's disease. The healthy brain is made up of billions of neurons in a really interconnected network. And in an Alzheimer's disease brain, a lot of the neurons have died. And so the connections have been lost. And in its place, what we see are the dead neurons containing neurofibrillary tangles inside the neurons. And in between the neurons, we see these amyloid plaques. And what happens is that this causes brain degeneration. It disrupts the neuronal network. So a lot of the brain functions are compromised.
All right, so let's look at a healthy brain, for instance. And what are we seeing here? The neurons are specifically, these are pathways that contribute to the brain's function.
So the brain is made of billions of neurons, and all the neurons are interconnected, and they make trillions of connections.
You talked about plaques and tangles, and those are some of the things that you see in the diseased brain.
Exactly.
And that's a much different, a big contrast compared to the healthy brain. Talk a little bit about that.
So in place of healthy neurons, in Alzheimer's disease, there's a lot of neurodegeneration, meaning that a lot of these neurons have died. And the brain as a whole literally shrinks in size if you compare Alzheimer's brain to that of a healthy brain. In place of that, there are two prominent hallmarks that we see, which are these neurofibrillary tangles inside dying or dead neurons, and also plaques, these massive proteins, amyloid plaques, that are in between the neurons.
What makes the good plaques go bad? Do we know that?
So what we know is that these plaques are composed of protein aggregates. That is, these proteins have come together to form in a particular morphology, particular form, in that they come together to form these fibrils. And that we know that both the plaques and tangles are composed of these tangles. What we don't know is what caused these proteins to undergo this transformation of forming these fibrils. So there are two questions that we still don't have the answers for. One is why do these proteins, in the population who does get Alzheimer's disease, undergo this process to form these fibrils, and also during the process, what causes the neurons to die? Why do these proteins start to lose their, their good structure to form this structure that are found in diseased brains? And what's the process of forming that? So what triggers it, and how does the process actually come about?
Do we know how we prevent them from losing their structure or their health?
We know what keeps a protein happy.
What's that?
In that
what keeps it happy and looking like that first slide we saw?
Right. So proteins, their functions depend on their structure. But these structures are very delicate. So you can think of the protein structure as a house of cards. So if any disturbance happens to any part of the protein, then the whole structure can fall apart. So it is that delicate. And actually our body spends a lot of energy to keep, for example, our body temperature at 37 degrees, keep the pH and water content inside and outside the cells exactly the same. We spend, like I said, a lot of energy maintaining that. So we know that under those conditions, proteins are happy. Once they destabilize, they can become very sticky towards each other, that they start to form these aggregates. If you think about, again, the analogy of house of cards, once one piece is removed or even bumped a little bit, then the whole structure falls apart. So what we're trying to do is find that trigger. So the scale that we're doing our investigation on is really on the protein level, which are between 1 to 10 nanometers in size. A nanometer is 1 times 10 to the negative 9 meters. And to change that scale to the scale of a brain, that's 12 orders of magnitude, so 1 with 12 zeros in terms of size. So we're going from how an individual molecule acts, which is at the heart of the problem, to how that interaction affects how the brain works, which is really what Alzheimer's disease is about. So the problem of studying proteins at the molecular level to kind of predicting the behavior of the brain, which, as we know, is about this big, we're dealing with a vast range of scale of sizes, and that's a complicated problem. What we're studying applies to not just Alzheimer's disease, but this class of amyloid diseases in general,
which includes Parkinson's and a host of other diseases.
And Huntington's disease,
Huntington's
for example.
disease. Okay.
So what we believe is that there are some commonalities with these proteins, in that they are, I guess, promiscuous in their interactions, that they have a higher tendency or propensity to interact with other structures in the cell. So what we're trying to explore is really kind of their abnormal interaction with the cell membrane that maybe can trigger the protein to what we call misfold, meaning that they go into a structure that's not a native structure.
Do they break the membrane of that cell they're in? And
that's a really good question.
Okay.
So the second line of investigation that we're trying to look at is to answer the question of toxicity. Why do these become toxic, or how do these proteins exert toxicity during aggregation? So what we're trying to do is looking at interaction between these proteins with the lipid membrane, and how that interaction affects the structure of the protein, and also how those interactions affect the integrity of the membrane.
What have we learned from this kind of research this far?
So what we've learned is that in the presence of certain lipid membranes, membranes made of certain lipids, they form the fibers much more quickly and at much lower concentrations. And the second thing that we found with their interaction with the membrane is that once these proteins start to aggregate, they interact more strongly with the membrane, and they can actually destroy the integrity of the membrane, essentially making the cell membrane that encloses the cell leaky. And once that happens, the cell can die. So your question about what can we do about it, what we envision is if the interaction between these proteins and the cell membrane can trigger or seed structural changes in the protein such that they undergo aggregation to form these fibrils, then if we block that interaction between the protein and the membrane, we can potentially prevent these fibers from forming.
Thank you.